Hello everyone. We are continuing with the series on biotechnology at CEC Gurukul, and today we take a session on advanced tools and techniques in which I, Dr. Charu Dogra Rawat, Associate Professor at Ramjas College, University of Delhi, would be discussing the techniques of AFLP, RAPD and DNA microarray. The learning objectives of today's session would be to understand the principle and procedure of AFLP and RAPD DNA fingerprinting and to understand about the DNA microarray technique. Now, talking about the DNA fingerprinting, we have already done multiple sessions talking about it, where we have seen that DNA fingerprinting is equivalent to DNA profiling, DNA typing, genotyping, genetic fingerprinting, as well as identity testing. These are synonymous terms, and it is a technique that simultaneously detects many markers in a DNA genome to produce a pattern unique to that DNA. So we have talked about it a lot and elaborated on the different kind of markers that can be used for DNA genome analysis, as well as when we use these markers and various techniques, then a unique pattern is produced for that DNA. And application wise, we can use it to even do the paternity testing, we can use it to genotype an individual, we can use it for identity testing, we can use it for knowing the different cultivars, etc. And we have discussed that in detail in our previous sessions. One of the major techniques that we have discussed is basically involving the restriction fragment length polymorphism. So DNA fingerprinting involves this RFLP markers, and these RFLP markers are basically when we digest the DNA with restriction endonucleases. Now again, if you recall, these restriction endonucleases are the DNA cutting enzymes that cut in between the DNA at particular sites called as the restriction sites. So they recognize particular sequences and they cut the DNA at that point. Therefore, they are called as nucleases, and because they do within the nucleotide chain, then therefore they are called as the endonucleases. For example, HIN3, its recognition sequence is given GTTAAC. It recognizes this sequence and makes a cut. So when we apply this digested fragments onto the nylon membrane and then we use a probe to detect it, then we can basically observe a unique pattern amongst various genomes. So this RFLP analysis is basically used to detect the change in the length of restriction fragments, and that occurs because all the genomes are not identical and they may have restriction sites at different places, and therefore when cut with a particular restriction enzyme can give rise to different length fragments. So that is why the name restriction fragment length polymorphism. So this picture just depicts the phenomena, and we have already discussed it again. So this is, for example, genome, and then when it is restriction digested, then when it is digested, then when you run it on a gel, then these kind of different restriction fragments are formed. It is then transferred onto the nylon membrane, and then probe, which is complementary to certain sequences or gene of interest, is used to hybridize these nylon membranes. Then what you can get is the signal, which is basically detect those fragments which are our gene of interest. So as we discussed already, that the DNA fingerprinting involves primarily the complementarity of DNA property, and therefore use it for the hybridization of the complementary DNA to the original DNA which we are analyzing. So here again you can see that the RFLP probe, or this is an RFLP marker, and when you use a probe, then it basically binds to these markers and therefore give rise to different length polymorphism. Now when you compare two different genomes, then in some cases these restriction sites may be disrupted, and therefore when it is disrupted, it may create or it may disrupt the restriction site, and therefore you get different kinds of fragments. So it can be used to detect a particular gene, as well as it gives a different pattern or a fingerprint, what we call the DNA fingerprint for different individuals. Then that can be compared. We had already discussed its applications in detail in our last session. Now talking about another technique based on this DNA fingerprinting is called as the amplified fragment length polymorphism. Now how is it different from RFLP? RFLP was restriction fragment. So basically it involved only the restriction digestion of certain fragments of the genome, and therefore it gave rise to a unique pattern. Now here the amplified word is used. So amplified is basically, that means that the polymerase chain reaction is involved. We again have a session on polymerase chain reaction, which is already discussed. So when we use different specific primers for identifying a particular region or gene of interest in a genome, we can actually amplify that part by using this PCR technique. And when we amplify it, then we also see that there are different kinds of fragments, different length fragments can be generated, which again gives a unique pattern that can be detected. So AFLP is a PCR based derivative method of RFLP, which sequences are selectively amplified using the primers. Now a subset of digested DNA fragments are amplified, so it does involve restriction digestion, as we will see subsequently. So the DNA is first digested, and then the specific primers are used to amplify or generate and compare the unique fingerprints for the genomes of interest. Now the significance of this AFLP is that it does not require prior information regarding the targeted genome. Now again, when we use probe, when we use hybridization, that we have discussed even in southern blot hybridization. So hybridization basically means that there is a binding of the complementary DNA sequences. So if we are using one of the sequence as a probe and we are looking for a target, then we should know somewhat about the sequence, therefore only we can design the probe and label it and then use it for hybridization purposes. But when we do not know, we do not have any information of the targeted genome, then this AFLP method can be used. Now you may ask that I am talking about amplification where we use specific primers, but then I am saying that you should not have an information regarding the targeted genome. So how is this possible? So we will see subsequently that how is basically this done. Another significance of this AFLP method is that it provides high reproducibility and sensitivity for detecting the polymorphism at the level of DNA sequence. So this is a very sensitive technique. However, one of the disadvantages of AFLP is that it requires a very pure form of DNA. So the crude DNA does not really give confident results, but you have to have very highly purified DNA and a high density DNA which can be used for AFLP analysis. Now what did I mean by when I said that what is the procedure how which it runs? So you can see, for example, this is a fragment and we digest this fragment with two enzymes. So one is an EcoR1 and another is an MSC1. This is a 6 base pair cutter and this is a 4 base pair cutter. So we cut this enzyme and we get a variety of fragments. Why? Because we may have EcoR1 digested fragments, we may have EcoR1 MSC1 digested fragments, and we may also have MSC1 digested fragments. So we have a large number of fragments. So at first what we do is we reduce these number of fragments before we can use it for amplification, specific amplification by using the primers of our specific for our gene of interest. So initially we digest it with these two enzymes, we get these fragments which are there, as you can see one of the fragment which is having EcoR1 on one side and the MSC1 on the other side, and then what we use are called as the adapters. Now adapters again we discussed when we discussed the recombinant DNA technology. The adapters are basically preformed sequences which contain the restriction sites. So now we are actually completing this restriction sites of EcoR1 and completing this site of MSC1 by using synthetic adapters. Now these synthetic adapters, however, this sequence is known to us because we have designed it. So we just align the aligning part to the DNA fragment will occur because of the restriction site that is present here. So we align these adapters and we add these adapters on the two ends which are there. Now we can do a pre-selection, because now we know the sequence. So even if we didn't know the sequence of the DNA fragment, we know the sequence of the adapter, and therefore we can use and design the primers for these adapters, and we can use it to amplify the fragment. So this is called as a pre-selective amplification. So in pre-selective amplification, this is basically primers are used and this is amplified. Then after that, this is basically the selective or specific amplification, because as you can see in pre-selective amplification, we use one primer, one nucleotide that we have added here, so that there is a little it binds to this particular sequence and still can get into the fragment of the gene of interest. Then we use specific primers, and in these specific primers what we are doing is we are making the primers and including, for example, 3 base pair of the fragment of interest. So this will again reduce the number of fragments of our gene of interest, because they will only bind specifically to our gene of interest because of the presence of these 3 nucleotides which are there. So mismatch is generally avoided at the 3' end. So this will give the specificity of the primer binding. So as you can see that the primers that are designed contain a core site, enzyme site, and the extension site. So for the core site, we use the sequences which we have included in the adapter sequence. The enzyme is basically the nucleotide sequences where what kind of restriction enzyme site we have used, and extension is basically the nucleotides of the gene of interest that we are adding. So this specific amplification will give rise to again reduce the number of specific fragments, and then they can be run. Then these fragments can be transferred just like for the other techniques to nylon membrane, and can be probed also, but this amplification pattern in any case would be distinct and unique for the particular sequences, and therefore comparing these AFLP markers, we can actually do the comparisons just like that we do it for the DNA fingerprinting. So this is another technique of AFLP which is very useful for the DNA fingerprinting method. Another very widely used DNA fingerprinting technique is termed as the random amplified polymorphic DNA. Now, as the name suggests, this is a random amplification. This is not specific to a particular sequence, but we use random primers to amplify the genome of interest at different places. Now, this random primers can bind to the complementary, and again we can change the stringency for the binding of these primers, and then we can amplify and get a pattern of the amplified fragments. Now, this is a most commonly used primary assays for screening the difference in DNA sequences of 2 species of plants, for example. RAPD consists of fishing for the sequence using random amplification, as I have already said. So what is done in this particular technique? For example, take the example of the plant genomic DNA. So the plant genomic DNA is cut and amplified using short single primers at low annealing temperatures, resulting in amplification at multiple loci. So what do we mean by these terms? Short single primers are the SSRs, which are again we used to amplify particular type of markers. So these primers are used and the low annealing temperature is used. Now, if you again recall what we discussed in the polymerase chain reaction, the annealing temperature is actually used for the optimum binding of the primer to its specific sequence. So this melting temperature, what we used, and there is a formula how we calculate the annealing temperature, is basically that the primer that we have used binds to its specific target. Now, when we use low annealing temperature, then there is a random primer binding that can occur, which means if there is a 10 base pair or 12 base pair primer, that not necessarily the 12 base pair should be identical in the complementary DNA sequence. Even if 5 base pairs are similar, it will result in the binding of the primer to the target size. So obviously this is a low annealing temperature, and therefore it results in the amplification at multiple loci, because the primers that we use in the genome are going to bind at different places because of its low specificity and low stringent conditions that we are using. Now then what happens after this amplification? A two-dimensional electrophoresis gel is run, and then it is possible to determine the change in sequence pattern by superimposing the two gels. So because we get different patterns and we run it on a 2D gel, then if we superimpose the two gels, we can actually identify the unique pattern and the unique fragments which may have occurred because of the change in the sequence, and therefore this change in the sequence can be very easily identified. Once the band of interest is identified, the gel is cut and the DNA is isolated and sequenced. Using this target, DNA from other cultivars can be assessed using other techniques such as AFLP or SSRs. So what happens once we have identified and detected a particular target sequence based upon the RAPD thing, then we can use these amplified products as probes to identify the target DNA in different cultivars. You know, it is basically a comparative genomic hybridization method where we identify and analyze different genomes by using, for example, one randomly amplified sequences from one of the cultivars, and then we can identify and then we can see where it binds, what region is present, what is the pattern, whether they are different or not, or you know, all these kind of analysis can be made based upon that. So even by RAPD technique, it can be clubbed by using this identification. It can also be used for other techniques such as the AFLP or SSR techniques. So this is how basically it is shown. So for example, this is a 7 chromosome thing, and when the randomly primers are assigned, then the randomly primers are binding to different regions here. So there you can see that there are different primers that are binding. Now again, important thing is that you can say that the primer is binding here as well, but it is not amplifying based upon the any other primer. Now PCR amplification is restricted for certain nucleobases only. So it can go up to like 1000 base pairs, but if it goes more than that, so the two primers which are randomly binding in opposite orientation and to the opposite fragments at a distance which can be amplified, it will only work for that. It will not amplify this using this primer and this last primer because they are on the other strands, but this distance is too much. So they are going to give rise to the amplified product of a specific size only. So you can see the different amplification products are obtained, and then this different amplification products can be run on the gel, and therefore it will give a kind of a pattern. Now this binding will be dependent upon the complementary sequences and will vary from the cultivar to cultivar, and even these RAPD primers can be used to hybridize with the other cultivars and can identify again the unique patterns, that whether they are present or what is their location. For example, this fragment might be present in this particular fragment, but this might be a little wider in some other case where it can be go and bind as a probe. So this is the RAPD technique that is utilized. Again, showing the same thing for two different cultivars. The primers are bound to the genomic DNA. As you can see, it is binding at different places, and it is only amplifying the product where the primers are binding in opposite orientation to different templates and at a particular distance. So for example, when we amplify this particular genome of interest, you get two different products, and when you run it on a gel, you will get two bands. Similarly, if you use another cultivar, then if you are using the same primers, they may bind at different other places, and therefore it is giving rise to a single product. And then when you run it on a gel, you will obtain that RAPD primers for two different genomes are giving two different patterns. So in this, it is giving binding at multiple loci, giving rise to two bands, and for this it is only binding at one band which is there. So this is again a very important technique. However, in this technique, the crude DNA can also be used, and therefore we compare the features of the RFLP as well as the RAPD method. So in RFLP method, as you can see, it is a codominant method. Now what do you mean by codominant? That the markers that are used, they are both basically expressed, or both alleles are expressed in the different genomes. So we can actually use it to identify homozygous as well as heterozygous individuals. However, when it is dominant, then it only shows the absence or presence of a marker. It does not show the different allele specificity of that particular marker. But RFLP is a codominant marker because the same region can be identified in both, but different length polymorphism will be given. Then as you can see, it binds to the multiple loci. It can, amount of DNA needed is 2 to 10 raised to the power 2 to 10 micrograms, which is used. You require the radioactive labeling for the probe, and the different types of markers that can be used is the sequence species specific probe can be used and other probes can be used, and the time required to do this exercise because it involves the complete southern blot hybridization. So time required is a little more. So these are the features of the RFLP. But if you compare it with the RAPD, then in the RAPD it is a dominant marker. So this dominant marker basically means it shows the presence or absence of a sequence, but it will not show that the same sequence is present in different allelic forms. So it cannot distinguish between the homozygous and the heterozygous, but all it can distinguish between is whether this this particular amplified product is present in one and not present in the other. The detection of multiple alleles of a marker cannot be done because it amplifies only the product where the specific primers are binding. The quality of DNA, the crude DNA can also work. As you can see, greater than 10 nanogram of DNA can also work. We do not require radioisotopes because we can directly identify the amplified product on the gel. And even in the capillaries, when we talked about the sequences, so we can run the amplified product, we can label it with different fluorescent dyes, and we can run and see the pattern and use it on capillary electrophoresis. So you do not need radioisotopes, you do not need any restriction enzyme digestion, and the type of markers that are basically used is basically the random primers that are basically utilized, and time required is less than 1/5 of the RFLP. So RFLP is a very elaborate technique. However, at this point I would like to mention one thing, that we are discussing, for example, different techniques of DNA fingerprinting. Now there is not any technique which is the best technique out of everything. That it is not that only this technique is the best and it should be used. Depending upon what we want to analyze and how much time we have, how much resources we have, and what kind of detection do we need to do, we choose these the technique out of the techniques that I am discussing. So there is no hard and fast rule that this is the best technique. RFLPs are used, AFLPs are used, RAPDs are used, and all different kinds of techniques of DNA fingerprinting which are given here, and the characteristics are also given here. They are basically used for that. So you can see that where the PCR primer is used in different kind of techniques, whether they are dominant or not dominant, what kind of DNA is utilized, what kind of how fast the technique is, all these different kinds of techniques. This is a comparative chart that you can see and appreciate that out of all these techniques we make a choice by comparing the different properties, what kind of hybridization is used, what kind of whether it is a PCR based technique or not. All these kind of techniques are basically utilized. Again, for this, as we see, these are further again more different properties, that whether they are utilizing the radioisotopes or not. So in PCR based methods we do not use radioisotopes, we can do away with that. But in restriction fragment length polymorphism, the probes are variously labeled, not only radioactively labeled, but they can be fluorescently labeled also. So we require that kind of a labeling. So what is the detection limit for all these different techniques, RFLP, RAPD, AFLP, even SSR markers. So these SSR markers are basically utilize the markers which are called as the SNPs. We talked about the SNPs, which are single nucleotide polymorphism, or there are short sequence repeats which are there. So short sequence repeats, the SSRs, are utilized to make these sequence specific primers, and then it can be utilized as well. So there are different methods and different employment of these different techniques of DNA fingerprinting that is basically used and utilized for carrying out the different purposes. Now, the different purposes that is carried out, we have already talked about the application part. When we are discussing the applications, then in the applications we utilize them for identity testing, we utilize them for as we said the genotyping, we utilize them for RFLP was utilized for even paternity testing, and depending upon whether they are multiple loci, whether they are codominant, whether they are require less time, whether they require a highly purified DNA or a crude DNA will work. So all these different things can be selected, the different DNA fingerprinting techniques can be chosen and employed in a biotechnology experimental work or generating of different biotechnological experiments. Thank you.